Thanks for all braving the elements and coming along today. Chris's wonderful talk there has actually given me the bug for a bit of audience participation. Hope you don't mind. So, starter for 10. Who can tell me who these two gentlemen are and what they've just done? Watson and Crick, thank you. What is it they've done? Thank you. Thank you. I like it, yes. Watson and Crick. And we're going to hear a lot about them this year. It's the 60th anniversary of their proposal of the double helical form of DNA. Rarely a week goes by now in the news we don't hear about DNA, whether it's being used to find horse meat in our burgers or identify the body of Richard III. But, you know, we can do more serious things with it. It's helped us make things like recombinant insulin. So, thanks to this, Watson and Crick got the Nobel Prize in 1962, and it's pretty much put them up there with the pantheon of scientific giants. And obviously, as was pointed out there from the floor, controversial, but we'll come on to that later. So, two scientific giants. Right. My next question. What's happening here? And who's this gentleman with the cricket bat? Ian Botham, thank you. Do we know what's going on? Headingley, summer 1981. England were losing the Ashes 2-0. Ian Botham came on, put in a legendary performance, became known as his 149 not out performance at Headingley. Turned the series round, England went on to win it 3-1. At which point, you may justifiably be sitting there and thinking, what on earth is the connection between Watson and Crick, the double helix, and Ian Botham? And I would say the connection is Headingley. Thanks to Botham's performance, Headingley, at least in cricket circles, has come to be associated with sporting giants. But I would like to make a case that Headingley also deserves international recognition for having had its fair share of scientific giants, and we've been hearing about two of those already this morning, Sir William Bragg and Lawrence Bragg. But what I want to tell you about today is that there's a third scientific giant who lived in Headingley. In fact, he lived just over the road from the cricket ground where Botham did his stuff. His name was William Asprey. He was a physicist. He was a student of Bragg. In fact, he called Bragg his scientific father. And when Asprey came to Leeds, he more or less laid the foundations for Watson and Crick. But he also did a lot more. And what I'd just like to talk about in the next 20 minutes or so is what he did. So, brief bit of biography. Asprey was born in 1898. He was the fourth son of 7 children, son of a potter's turner, in the vicinity of Stoke. And he excelled academically from an early age, won a place at Cambridge, went there, and he became very interested in crystals while he was at Cambridge. And he wanted to carry on that research when he finished. Well, at the time, if you were interested in crystal structure, there was only one person to go to, and that was Sir William Bragg. Sir William Bragg had left Leeds by this time. He was in London, first at UCL and then at the Royal Institution. And this is where Asprey joined him. Sir William Bragg had by then recruited a group of kind of eager, bright-eyed young scientists who were keen to make a name for themselves in crystallography, and one of them was Asprey. And it was at this time that Bragg, we heard earlier, Bragg got his Nobel Prize for using X-ray crystallography to look at compounds like salt, sodium chloride, inorganic crystals. But when he got to London, Bragg set himself a new challenge. He began to wonder whether X-ray crystallography could tell us anything about substances in living things. And one particular substance has caught his eye. It was this stuff, wool. Now, it might not seem to be the most interesting subject for scientific study, but there was a reason why there was interest in wool. Give you a bit of historical context. It was the aftermath of the First World War, and the German economy was devastated both by the cost of having to fight the war and the burden of the war reparations repayments. So the German government was looking for ways to get the economy going again. It all sounds very timely stuff this week. One of the ways we wanted to do that was to support science that had a very practical industrial application, and one such science was the science of textiles. So there was a lot of work going on in Germany on textiles, and the knock-on effect of that was to stimulate work on textiles in Britain, and wool was the main raw material for textiles in Britain. And Bragg felt that this was an area where Asprey could really make a career for himself. But the place to do that was not down in London. The place to do that was at the centre of the wool industry, which was up here. Leeds and Bradford were the centre of the wool industry. If you go down the road to Kirkstall Abbey, ever since the Middle Ages, the monks at Kirkstall Abbey had been selling the wool from their sheep abroad. And by 1820, I think it was, Leeds actually had the biggest wool mill in the world, which is still there today at Armley Mills Industrial Museum. So in 1928, Asprey came to Leeds, the aim being to use X-ray diffraction to study wool fibres for the local industry. Wasn't a move that he was particularly enthusiastic about at first. Wool really seemed a bit like a dead end. But he came up here, he began his work with the camera we can see here, which is on display up in the Centenary gallery, working in what's been called very much a strings and sealing wax tradition, because rather like William Bragg building his own spectrometer, Asprey had to build this camera. This was an age when the scientist was very much a craftsman as well. And so he began using X-rays to study wool, which had been pretty much written off as biochemically lifeless. How wrong people were, because through his work on wool, Asprey was able to answer a fundamental question which had perplexed biologists and chemists for a long time, and that is, what are proteins? Now, we hear a lot about DNA today, but it's actually proteins are very much the Cinderella molecule of the cell. Proteins are the workhorse of the cell. Protein hemoglobin carries oxygen around the body, the protein in the back of the eye, rhodopsin, converts light energy into electrical energy. There are protein hormones like insulin and countless digestive proteins like enzymes. So proteins are the workhorses. But at the time, nobody knew what proteins actually were. It was known that they were made of up to 20 different smaller compounds called amino acids. But nobody knew how these amino acids came together to form proteins that could perform this diverse array of functions. Well, it's with his work on wool, Asprey provided an answer. Because when Asprey studied the keratin fibre proteins in wool with X-ray diffraction, what he was able to show was that the keratin proteins were long chains of these amino acids joined together. Why was that novel? Well, we think of molecules as being very small things. Carbon dioxide, 1 carbon, 2 oxygens. Water, 1 oxygen, 2 hydrogens. What Asprey showed with his work on wool was that in biology, at least, molecules can be huge. They can be juggernauts. But more importantly than that, they can do something interesting with these giant molecular chains. Because what Asprey found when he did X-ray diffraction on the keratin fibres were, he found that the chains, the huge great amino acid chains of keratin, could actually change shape. They could take one of two forms. They could either be a compacted, coiled up alpha form, which you call alpha keratin, shown on the far left, or they could stretch out like that. So I brought the slinky along just to give you an idea of what's going on there. They could be stretched out into the beta form, the extended form. Why was that significant? Well, it actually explained a key property of wool, the property that made it attractive as a textile raw material. It's elastic, it stretches. For the first time, Asprey was given an explanation of a gross biological, macroscopic property in terms of changes at the molecular level. And this pretty much formed for him the definition of a new science. Because for him, living things could be understood in terms of these giant chain molecules and how they change shape. And for him, that formed the new science of molecular biology. Prompted one of his colleagues there to come up with this witty little limerick: amino acids in chains are the cause, or so the X-ray explains, the stretching of wool and its strength when you pull to show why it shrinks when it rains. That brought Asprey international fame, these findings on wool. It really put him on the map scientifically, and he began to broaden his work and look at other protein fibres in biology. He looked at, say, the flagella, that's the structure, the whip-like structures that propel bacteria through water. He looked at muscle proteins, looked at the keratin proteins in human hair. And he was a great lover of classical music, so it was to that he turned for an analogy. He said that the fibrous chain molecule is nature's chosen instrument in the symphony of creation. It's a nice metaphor. It's even more timely, actually, that the picture on the far left, the X-ray diffraction picture of human hair, the hair sample for that actually came allegedly from the head of the composer, Wolfgang Amadeus Mozart. Quite nice. How that came to Leeds, that's another story. But basically, Asprey's work on biological fibres put him on the map so much that the Nobel Prize winner Max Perutz, who went on to win the Nobel Prize for solving the hemoglobin structure, hailed Asprey's lab at Leeds as the X-ray Vatican. Another kind of timely reference there. But Asprey didn't confine his X-ray work to proteins. He looked at the huge, great, complex sugar molecules on the surface of cells, and there was a third class of biological fibre as well. It was white and stringy. It was isolated from the thymus gland in calves, so they called it thymonucleic acid, but today we know it by the much more familiar name DNA, deoxyribonucleic acid. And in 1938, Asprey was sent some samples of DNA, and his PhD student there on the right, Florence Bell, took X-ray pictures of them. So the very first X-ray pictures of DNA were done here at Leeds in 1938. Here's one of Florence Bell's pictures. It might just look like a blur of spots. But from that, Asprey and Bell came up with the very first structural model of DNA. Again, it was a little bit like the situation with proteins. People knew DNA was made of sugar, ribose, phosphate, 4 chemicals called bases. Nobody really knew how those components came together. But Asprey and Bell's X-ray work showed that they were joined together in a long chain, and that the bases stacked on top of each other to form what they called a pile of pennies. And from that work, they were even able to make a key measurement. They could actually measure the spacing between adjacent bases. We know today that their model's wrong. We know that DNA is 2 chains, but it doesn't mean the model's worthless, because that measurement between the bases was crucial. In fact, James Watson later acknowledged that it was Asprey and Bell's measurement of the distance between the bases that gave him and Francis Crick a key foothold when they actually began building their first model of DNA. And I should say at the time, nobody really knew what DNA did at this time. People knew that a molecule must be involved in passing on biological information, but the smart money was on proteins being that molecule. And the reason for that is, if you're going to pass on biological information, you need some way of encoding masses of variation. Proteins seem to be the best way of doing that because, as I said, proteins are made of up to 20 different amino acids, so you've got a lot of capacity for variation there. DNA, by contrast, is just made of 4 bases. It seems much more limited. If you want a helpful analogy, think of an alphabet. How many more words can you make with an alphabet of 20 letters than you could with an alphabet of just 4 letters? So DNA seemed to be a pretty dull molecule, actually, and most people had written it off as just being a structural molecule, like a kind of molecular girder. I say most people. Asprey wasn't one of them. The reason he wasn't one of them was he took notice of an experiment that had been done in 1944. There was a Nobel Prize winner, Josh Lederberg. He once said 1944 was a pivotal year in human history for 2 reasons. First reason, the successful landing of the Allied forces on the beaches of Normandy to liberate Europe on June the 6th. Second reason, an experiment done by a shy retiring clinician called Oswald Avery in New York in 1944 that some biology textbooks now hail as Avery's bombshell. What Avery did was, he was working on the pneumococcus bacterium, shown on the right. Pneumococcus comes in 2 types, disease-causing, non-disease-causing. And Avery showed that DNA, and DNA alone, is enough to transform the non-disease-causing into the disease-causing. Asprey picked up on his work. Asprey called Avery's work one of the most remarkable discoveries of our time because it obviously meant that whatever DNA was doing, it was an awful lot more than just being a molecular girder. He wrote to Avery and asked for a sample of the DNA. Don't think Avery ever actually sent it. As I say, he was pretty shy and retiring, kept himself to himself. But, nevertheless, Asprey's DNA work carried on. In 1951, his research fellow, Elwyn Baton, shown there on the far right, took some new photographs of DNA. Now, I hope you can clearly see the spots formed by the scattered X-rays there form an X pattern. Chris has already been talking about Rosalind Franklin's famous photograph of the X pattern, which we see here. James Watson always said when he was shown this photograph, Rosalind Franklin's picture, and he was shown it without her permission and without her knowledge, he said, my jaw dropped and my pulse began to race, because Watson knew that only a molecule that was helical in shape would diffract the X-rays to give you that X pattern. And Watson acknowledges that that picture was probably the vital clue for him and Crick in solving DNA. If you walk down the Strand in London, where King's College is, there's a plaque that hails that picture as one of the world's most important photographs. But the intriguing thing is, 2 years earlier, we've had pretty much the same photograph here at Leeds from Elwyn Baton. In other words, we had the clue to DNA right in front of us, but as we all know, history didn't work out like that. Why it didn't, we could maybe go into if there's time at the end. Because I actually think it would be a mistake just to focus on Asprey and DNA. Right? His legacy was bigger than that. His legacy was that you can understand biology in terms of big chain molecules and how they change shape. And today that explains for us how hemoglobin works, how rhodopsin works, how insulin binds to its receptor, how digestive enzymes work. But just to wind up, he went one step further. I want to show you this newspaper cartoon. Appeared in the Boston Globe in 1977. Shows a stereotyped mad scientist racing into his lab being greeted by this host of genetic mutants. The cartoon is an example of the kind of fear that was being felt in the media at that time about the advent of genetic engineering. In the early 1970s, scientists in the US had discovered restriction enzymes, which meant they could chop precisely a piece of DNA out of one organism and stitch it into the DNA of another. So people began worrying, what if you take a piece of DNA that causes human cancer and you put it into a cold virus? But it was pretty quickly realised there were some industrial implications to this as well. Herbert Boyer, professor of biochemistry at Stanford, took the sequence of DNA that encodes human insulin, stitched it into bacteria, so now you could get bacteria churning out industrial scale quantities of human insulin. And that's now used in the treatment of diabetes and been of untold benefit, and it landed guys like Boyer on the front page of Time magazine. In other words, for the first time we could deliberately, artificially manipulate these giant chain molecules and use them, put them to industrial use. Now, that's the standard telling of that story, but here at Leeds, we like to think there's an alternative. There's an alternative history here. We like to think that the idea you can deliberately manipulate chain molecules and put them to industrial application didn't start under the sunny blue skies of 1970s California. It began more under the rainy grey skies of 1930s and 40s Leeds. To explain why, I'll show you another newspaper cartoon. This one here, Yorkshire Evening Post, 1944. Gentleman in an overcoat, little birds are landing on his overcoat, pecking them. What's that about? Late 1930s, Asprey was doing X-ray diffraction not on fibrous proteins, but on the other kind of protein, globular proteins, where the chain's coiled up into a ball. He's got them out of seeds. And he had the idea that you could use chemicals to disentangle the globular protein chain and re-engineer it into a fibrous protein chain and use that as a raw material for textiles. In other words, you could basically take seed proteins and turn them into an artificial textile fibre. And ICI liked that idea so much, ICI bought the patents from him and began actually to set up a pilot industrial plant in Scotland, producing an artificial fibre that was produced from re-engineered monkey nut protein. And as proof of the pudding, they gave Asprey an overcoat that was basically made from this monkey nut fibre to wear, and he sported it at various lectures. So as I say, we like to think here at Leeds that this idea of manipulating, deliberately manipulating large chain molecules and using them for industrial purposes actually had an alternative origin. So just to finish, if you read standard stories of the story of the discovery of DNA and things, Asprey's pretty much relegated to a peripheral figure. He doesn't always get the most gentle of treatments. For instance, Brenda Maddox in her biography, Rosalind Franklin, described him as an unpersuasive man working at an unfashionable northern university. Ouch. Leeds Civic Trust thought differently. And a couple of years back, they honoured him with this blue plaque, which sits on his house. We began the talk in Headingley with Ian Botham. Seems like a good place to finish in Headingley. The plaque sits on his house, which is just over the road from the cricket ground where Botham did his stuff. So just finally, I'd like to thank everyone who's been involved over the years with me in doing this work, the Museum of History, Science, Technology and Medicine at Leeds, the staff of Special Collections, Prof Greg Raddick here at Leeds for pointing me in the direction, and all the staff and students in History and Philosophy of Science, Jim Garrett's from Thackeray Museum, Dr Keith Parker, a former colleague of Asprey, Professor Monty Lazowski, friends of Thackeray Museum, and a very special thanks to the members of the Asprey family who've been really supportive through all this. And thanks to you all again for turning up. And just one final note, the Science Museum in London is doing an online vote for the top 100 British innovations. X-ray crystallography is in there, right? And on Wednesday it was in third place, I think. The number one slot was occupied by the Mallard and then the Mini Cooper, right? We've knocked them off the top slot. We currently hold the top slot and we'd like to keep it there. The poll closes tomorrow. So if you all go out and cast a vote for X-ray crystallography, that would be great, thanks. Thank you.